Dr. Adam Cobb is our next speaker, as well as our very own Soil Food Web School science communicator, as well as an agroecologist. Adam's passion for agriculture emerged in 2008 during 3 months volunteer in New Zealand on organic farms. From there, his time in graduate school cultivated a broad vision for restoration of living soils, as well as utilization of research and community engagement to address current and emerging global food production challenges. After completing his doctorate at Oklahoma State University in 2016, he received a competitive NIFA fellowship for his postdoc. During the past 5 years, Adam authored or co-authored 20 research publications focused on agroecology or plant microbe symbioses. Today, Adam works as the science communicator at the Soil Food Web School, and he and I often collude to advance the unanswered questions for how we can better understand, teach, and work with the soil food web. Today, he will share his insight on one particular group that he featured highly in his research, mycorrhizal fungi, and how we have allies in the soil. Take it away, Adam.
Hello, my name is Dr. Adam Cobb, and today I'll be speaking to you about one of my favorite topics, which is the allies that live in the soil right beneath our feet. I'm a content creator and science communicator here at Dr. Lane's Soil Food Web School, and since joining the team, I've really been putting together ideas from my previous life as an academic researcher with the amazing things that Dr. Lane has taught me here about soil regeneration. I'd like to start by giving you just a little background into me and my passion for agriculture, which really began when I went on an extensive backpacking trip around New Zealand and volunteered on organic farms there. I came back and really decided that I wanted to take a new career path into the agricultural space. And eventually I earned a master's degree and a PhD from Oklahoma State University. During my PhD, I studied the interactions of plants and soil microbes. In the lab that I was part of there at Oklahoma State was actually more of a grassland ecology lab. And so later on in my career, I had opportunities to join in work, for example, in Botswana in 2018, where we were looking at ideas related to drought tolerance in grassland species. And the other big event that shaped my thinking was a program I did with a number of food producers for 2 years across all parts of the state of Oklahoma and several national locations. And we actually ended up going for 2 weeks to Chile to learn about different systems of agriculture and especially conventional food production systems. And then I moved here to Oregon in the summer of last year, and it's been wonderful. My dog and I really enjoy the Pacific Northwest. The really neat thing about my interest in soils developing, around 2009, 2010, I really got excited to invest myself in the science of soils, is that the world came along with me. In the last decade, we have seen so much more focused attention on defining soil health, understanding how to regenerate soils. It's happened in national organizations, it's happened internationally through the UN, this real growing, emerging movement for soil health. And that's really because the soils in many places are not in great shape. And as we look back even to what was happening in the Dust Bowl in the US, we see that, this idea of the soil relating to national security through food security. We can go even further back in time, and we can think about the fall of ancient civilizations such as Babylon that were really, at least partially, the destruction of their soil ecosystems and resource base explains why those mighty civilizations fell apart. And so I mentioned that I have spent some time studying grassland systems. One of my favorite places in the world where I took this picture is at the Tallgrass Prairie Biological Research Station, and that's in the Central Great Plains of the United States, in Kansas, and it's one of the most amazing ecosystems that I've ever observed. If we think about the fact that historically there were 40 to 60 million bison that were roaming across the plains, and that there was very little management other than maybe fire, intentional fires, and very few inputs into these systems, and yet they continue to be one of the most productive ecosystems on the planet. And I really didn't understand when I was visiting the Tallgrass Prairie, why is the system so productive? What makes it, how could we bring these lessons into our other agricultural or food production systems? And then, as I said, when Dr. Elaine helped me put these pieces together, many of these components I had studied in isolation, but when she helped me see this holistic soil food web concept, I said, that's why the Tallgrass Prairie is such a magnificent ecosystem, is that these relationships are functioning. There's so much diversity. Sometimes we see 15 or 16 species of plants per meter squared in the Tallgrass Prairie. And then as we look below ground, there is this healthy community of protozoa, nematodes, and fungi, bacteria, and arthropods that really explain why that the nutrients in that system are cycling, and how that contributes to such a massive amount of grassland productivity. And so I really have found this quite an elegant answer to the questions that I had. When we look even broader than soil ecosystems and think about the way that human communities are impacted by relationships between agriculture, the environment, and nutrition, we arrive at this model or this philosophy that has been previously published in the literature that's called eco-nutrition. And it really explains to me a lot of the vicious cycles that can be observed in many developing countries, in many rural communities around the world, that are stuck in a cycle of degradation, especially driven by the loss of their productive soils. So starting with agriculture and our understanding that agricultural practices can influence the soil. It can either cause improvements to the soil's condition, or it can degrade the soil. In this example, the degradation of soil by agricultural mismanagement can lead to a problem in the wider environment, because as people are not able to produce as much food from their farm systems, there's additional pressure put on the natural areas around these communities. As desperate people who are living in poverty and dealing with hunger and malnutrition, they may be poaching wildlife. They may be cutting down trees illegally to sell those products so that they can buy food. They may be overfishing, right, from their local streams. And so there's a connection between the food security element and the environmental element. And overall, that leads to a declining yield, the loss of those productive soils, not just on the farm, but a decline of the whole food base around the community, which can get us into a horrible position in terms of nutritional deficiency, human nutritional deficiency. And that has this feedback effect on the cycle, because if we see children especially that are raised with limited nutrients, I'm thinking especially about things like protein, zinc, and iron are some of the major malnutrition issues around the world, then that causes diminished human capacity. Those children may be physically smaller than they would have been. They may be stunted. They may be mentally stunted as well. And so it creates a generation that is even less set up to manage agricultural systems effectively. And that's where we get this vicious cycle. The good news is that if we can improve our agricultural practices such that we get a regeneration of soils, that can enhance not only the on-farm environment, but it can take pressure off of those off-farm natural environments that I mentioned. And there's great examples of this around the world where projects that really effectively improved the production of farmers, the sustainable production of their agroecosystems, ended up causing a lot of other benefits to trickle out into their local environments. And so you can actually see a yield gain or a food resource-based gain when you build back soils. And that can lead to a nutrition surplus in the community, which then has this feedback effect of increasing human capacity, that we can have children that actually are have more capacity mentally and physically than their parents' generation. And so then you go into a virtuous cycle, right? Why am I setting all of this up? Because we have to face the fact that the Food and Agriculture Organization of the UN and other groups have identified that the loss of soil health around our planet is a huge looming crisis for our society, and that more communities run the risk of falling into those vicious cycles described by the eco-nutrition framework. We can look forward and talk about how many harvests are left before we run out of topsoil or how rapid the decline is, but we can also look backwards to what the historic productivity of a lot of these systems actually was. And in that case, we see that around 30% of the capacity of soils has been lost in the last 100 years. That even farms that we're producing large agricultural commodities from, that the yields historically would have been higher than they are now. And there's a lot of factors that go into that, but the loss of soil directly through erosion is a major factor. Now, if we overlay the parts of the world that struggle with soil loss, the places where it's happening most rapidly, in many cases we see that they're the same parts of the world that are struggling already to create adequate calories and nutrition for their populations. And in fact, hunger and malnutrition, this map is demonstrating just iron deficiency, anemia caused by iron deficiency, is one of the most prevalent problems in the world. Over 2 billion humans suffer from iron deficiency. And it also has a huge effect. Nutritional deficiencies have a huge effect on health. Globally, hunger and malnutrition are considered to be the #1 risk factor in loss of life and loss of quality of life. So really, this was the question that captured my attention during my PhD, is how can we link soil ecology to human nutrition and then leverage soil ecology to improve human nutrition? In order to stage this conversation about soil ecology and human nutrition, I want to work first through plant nutrition pathways, of which I have identified primarily 3. The one that I was most familiar with before coming to Soil Food Web School was actually the pathway that most modern chemical-based agronomy is focused on, which is a very inefficient pathway, but it's the idea of dumping soluble nutrients in the form of synthetic fertilizers near to the plant, right to its root zone, because of the way that plants can naturally take up nutrients that are near their roots. The problem is that we know that something like 60% of the nitrogen and phosphorus that we load into these agricultural systems just wash into our rivers and streams and cause environmental problems. And so I was very excited when I came to Soil Food Web School to learn about this alternative regenerative pathway of plant nutrition that's very dynamic, where we have predator organisms that are feeding on prey organisms close to the root systems of our crops. And those predators are excreting the extra nutrients that are in their bodies because of all of the bacteria and fungi that they're eating. They end up generating a lot of extra nitrogen and other resources, and they excrete those. And so there's these dynamic bursts of nutrients all around the root system. That's a rapid cycling, just-in-time kind of phenomenon, instead of that dumping that load of fertilizers all on at the beginning of the season. Now, when I was in graduate school, I primarily researched a mutualistic microbe. And so this is the third pathway that I kind of refer to as agroecology, but it's really about the symbiosis between most of the plants that we grow in agriculture and specific partner organisms. These relationships often exist in large evolutionary timescales, millions of years. These partnerships have developed. One of the most famous that we could talk about would be rhizobium, but that's not the example I'm really going to go into today. As the title of my talk is about the allies in the soil, I'm going to introduce my favorite tiny ally, which is arbuscular mycorrhizal fungi, or I'll probably call them AM fungi throughout this talk. AM fungi are one of the most abundant and one of the most meaningful plant mutualistic partners in the world. So let me give you some important background on AM fungi and why they're such amazing allies in the soil. AM fungi partner with the vast majority of our agricultural crops and many of the grasses in our pasture systems and trees and all kinds of things. It's estimated something like 80% of the plants in the world can form a relationship with AM fungi. And this illustration demonstrates that the exciting thing here is that these fungal structures can connect directly into the roots of plants. And there's actually an exchange of nutrients that happens. The plant has typically excess carbon in the form of sugars. And the fungi gather some of the soil nutrients that are most limiting to plants most often. Examples like zinc, ammonium, and phosphorus. They can also help plants absorb more water from the soil system. So it's a matter of extending the reach and the total volume of soil that can be effectively mined for the resources that the plant needs. But as the AM fungi reach out from that plant root system and build their biomass, their structures in the soil, they also entangle soil particles. So you end up with smaller pieces or crumbs of soil becoming larger aggregates. And that's very important to the stability of soils. We know that AM fungi can help reduce erosion. We know that they can increase aeration in the soil, that they can improve water infiltration in soils. So they have this effect on the entire soil ecosystem when they're operating in partnership with plants. The good news is that AM fungi exist on every continent except Antarctica, perhaps, and they're abundant and they're very beneficial. In terms of abundance, we can talk about the total biomass in the soil, and of all the microbes in the soil in grassland and many agricultural systems, AM fungi represent about 50% of the biomass. Another way that I like to visualize it is that if I had just a teacup of really healthy soil, like maybe soil I collected from the Tallgrass Prairie, that you could go in, if you had the patience, and stretch out all of the AM fungal hyphae in a chain, end to end to end, and it would stretch across 30 football fields. It's not, however, just end to end to end in nature. It's in a network. It's jumbled around in the soil. It's holding those pieces together, as I mentioned. And to give you an idea of the benefits of AM fungi, one of our former students, Caitlin, is holding here root systems of, in this case, switchgrass, which I'll talk a little bit about our switchgrass research later. And we have a method of keeping everything in the soil consistent, except knocking out AM fungi from the soils that we grow plants in. And so in the greenhouse, she was able to grow switchgrass plants that did not have AM fungi and switchgrass plants that did, side by side. And then we can compare how much more of a robust root system that the plants were able to develop when they had their AM fungal partners. Now for the bad news. Research that I've conducted in Zambia, actually at my friend's Peace Corps village that was about 20 kilometers away from the border with Malawi, would suggest, and put together with many other papers around the world, would suggest that we're really going in the wrong direction in terms of the abundance of AM fungi in our agricultural systems. I assessed some specific biomarkers that correlate very well to AM fungi in phospholipid fatty acids and neutral-lipid fatty acids. And we used a local grassland in this part of Zambia to represent a kind of control for us, or give us an idea of what the potential for AM fungal biomass was in the midst of like a thriving, healthy ecosystem. Compared to that, any of the farms that we visited, and there were a range of practices, some more regenerative than others, going on in these farms. Smallholder farmers, usually a hectare or less of land that they were managing. In many cases, you're talking about management through like oxen-drawn equipment. But what we saw in the data was almost a 50% reduction in AM fungal biomass. So this loss of AM fungi around the world makes us very nervous because we know how beneficial it can be to plants. In another example, we've knocked out the AM fungi in these alfalfa plants, and they were grown for the same length of time as the ones in the background that had AM fungal partnerships. And I don't want to lose all of that potential productivity as we see AM fungi diminished through agricultural mismanagement. And so really it begs the question, how can we unleash this symbiosis in the larger context of our food production systems? In order to help us dive in to answer that question, we really needed to develop the concept of what a model plant or crop would be. And we landed on sorghum bicolor, which is sometimes called milo. It's just referred to as sorghum in many countries around the world. It is one of the top 5 most abundant grain crops on the planet. And it's very drought tolerant and efficient. For example, as compared to corn, it is about twice as efficient. You can use half the fertilizers and half the irrigation, even in a conventional system, to get a good yield out of sorghum. But it's also related to many of those important crop species and grasses that we use in our food production systems. For example, corn, which is very genetically and physiologically quite similar to sorghum. Although sorghum has got a simpler set of genetic components than many of the other crops that we would be comparing it to. And so it really provides us this ideal model for the functional genetics of the plant, that we can observe what changes in gene expression in these plants and get have a faster route to discover the function and the ways that we could change the genetics to get the effect that we want in the world. It's a great model crop. And this is really where my PhD research started. I worked with a group from a USDA Agricultural Research Service lab, and we wanted to compare sorghum genotypes across this genetic gradient, especially comparing very highly developed, like commercial varieties of sorghum back to more ancient varieties, or things that we might call like a landrace or an heirloom. Now, because I'm going to talk about the effects of breeding, I want to make this caveat. I believe that, by and large, crop breeders who've given a lot to us in the world of agriculture. They've worked to improve the yield, things like disease resistance. They've brought a lot of benefit to the table. Historically, though, not a lot of attention has been paid to the below ground world of plants, and especially how they interact with and signal chemically with their long-term microbial partners. And so we think that there's a chance here that our breeding practices, remember, most breeding programs are set up where they're giving a high rate of fertilizer and maybe a lot of supplemental irrigation to the crops that are being bred, which we know can disrupt the whole soil food web and can absolutely decouple that relationship between mycorrhizal fungi and host plants. So by breeding in that situation, have we accidentally reduced the investment of our crops in this kind of relationship? This is the big question. And what I found in my research is that that's exactly what happened. Or we have an indicator that that is what's going on. So to orient you to my graph here, I have these two groupings of sorghum. The green bars represent those inbred genotypes, or those, like I mentioned, landrace or more ancient varieties of sorghum, as compared to these commercial hybrids that have been through a lot more of a selection process. On this side, I want to talk about the growth of the plant, and then also these bars at the top represent the production of the grain, because we were interested in human nutrition, so we wanted to look at the actual quality of the grain. When we do fertilization of any of these plants to the style that is typically done in breeding programs, we see a fairly equivalent productivity. However, when these different varieties of sorghum are grown in soil that's got relatively low concentration of phosphorus and nitrogen, these older landrace varieties produce significantly and substantially more vegetation and grain than those modern commercial hybrids. And when we dig into the below ground data, we see that the root area that's colonized by AM fungi on average is 3 times higher than the total root area that's colonized in the hybrid genotypes. We think that these things connect. They definitely tightly correlate or link. So when we put the root colonization that we observed in our plants on one axis, and then various minerals, in this case phosphorus, as well as protein, along the other axis, we see this tight relationship that those inbred genotypes that I mentioned, those more ancient, less modified varieties, were colonized more heavily by AM fungi, and there's more total grain and more, in many cases, concentration of nutrients in the grain. This was true for zinc and iron and many other examples in terms of the minerals that we assessed. So where does that leave us? Well, my take-home message is that we saw that varieties of sorghum that can more effectively partner with AM fungi could end up generating about a 30% gain in total human nutrition, right? That if you were planting a field of one of these less effective varieties of sorghum that didn't partner with AM fungi and comparing it to a field of plants from one of those varieties that was a good partner with AM fungi, that you would see a massive difference in the protein, zinc, and iron that you could actually harvest in the grain out of those two fields, as well as the total yield of grain. So the place that we would go next is to conclude we've probably gone the wrong way historically. Our breeding programs have probably inadvertently cut off some of the capacity of our crops to effectively partner with soil microbes. In this case, AM fungi is our example. If we've gone the wrong direction accidentally, it suggests that we may be able to go the right direction intentionally. And so that was the next question that we came up with is, could we breed crops intentionally to enhance mycorrhizal benefits? And it just so happens that there at Oklahoma State University, we had a robust switchgrass breeding program already in operation, and that they had a little bit of a different philosophy that I'll get into later in terms of the breeding style. So we're really tracking two main types of assessment here when we compared the growth of these different varieties of switchgrass. We wanted to know the mycorrhizal growth response. How much did the plant grow? How much more did the plant grow when it had access to AM fungal partners? And also the phosphorus response, because phosphorus is such an important mineral nutrient. And it's one of the ones that AM fungi we know can really help plants to acquire. There's actually a well-established way in the scientific literature for us to track the influence of AM fungi through these kinds of growth responses or nutrient responses. My picture is an example more recently that I had from a sorghum trial, and we have mycorrhizal plants over here and non-mycorrhizal plants over here. So we can actually subtract the biomass of the non-mycorrhizal plant from its corresponding mycorrhizal plant rep. And then we can divide the total by the non-mycorrhizal plant's average growth, multiply by 100, and we get a percentage of increased growth that we can attribute to the influence of AM fungi. There are two main takeaway messages that I want to get into related to the outcomes of this trial. The first is that all the switchgrass varieties that we assessed, when they were grown with AM fungi, grew a lot bigger, like 1,000% to 3,000% bigger. It's a substantial amount. But the other thing I'd like to point out is that these varieties that were produced from this program were based off parent lines, Canlo and Alamo. And because this breeder was, we believe, because this breeder was managing this breeding system differently and using a lot less fertilizer and using almost no supplemental irrigation during the course of the breeding, that the plants actually invested more, the offspring lines invested more in this relationship with AM fungi. And this is why we saw a significant and substantial increase in their mycorrhizal growth response. It was a similar pattern for the phosphorus response. So when we actually assessed the tissue quality of these varieties of switchgrass, we again found that even the parent lines had a substantial amount more phosphorus in their tissue when they were grown with AM fungi compared to when they were grown without. But that their offspring lines were significantly more concentrated or more abundant amounts of phosphorus in the tissue. And that's really remarkable. It suggests that there's something in these breeding protocols that has gone the opposite direction that we observed with sorghum. Now, currently, I'm actually finishing up a new project with grain sorghum that was funded out of USDA's NIFA postdoctoral fellowship program. And this is work that I conducted at OSU just a year before I came to Soil Food Web School. I actually grew 50 genotypes of sorghum this time, and we found many remarkable genotypes that had a robust response to AM fungi, and some that didn't have a very robust response. They did not grow much bigger because of partnership with AM fungi. And so we're actually going in and using molecular tools to look at the gene expression in these different varieties, and also to look at which taxa of AM fungi the plants are associating with, so that we hope to unravel, using those molecular tools, additional information about how our breeding programs have changed the relationship of AM fungi and grain sorghum. This is really focused on improving the breeding tools that we have available to us. So if historically we haven't been able to focus on these below ground relationships, we want to bring something new to the table to help breeders do that. So putting this all together, in my last paper, we compared traditional plant breeding, which is very focused on just the genome of that specific crop species, to an emerging concept called hologenome breeding, which is where we consider the plant, but also the context of its community of microbes, especially those mutualistic partners like AM fungi that are growing together. And we assess the way that the ecology of the microbes in the plants, and the way that soil amendments in the system, and the way that the management of the system can influence the outcomes. So we end up with either we go in the wrong direction or we go in the right direction. What's remarkable, since we published this paper before I came to Soil Food Web School, is that I got here and I discovered that considerations of soil microbial ecology, considerations of soil amendments, and considerations of agroecosystem management are really at the heart of what we're doing at the Soil Food Web School and what we're training producers to do. So I see a lot of compatibility between the work that I had previously done on crop breeding and what our mission is here at Soil Food Web School. And I'm going to put that together in using a metaphor of an arch. So what I hope that you see here is the tops of columns and the stones that go into the arch, and then this very important piece that you would call the keystone, the element of the arch that really holds it all together and allows it to bear weight. And what I have found is that traditionally, in agronomy, the soil environments and the management of the farm are the main considerations. How we do inputs and the timing of our crop systems, how we manage or how we deal with the effects of climate and stress in the system. This is where a lot of the focus has traditionally been. And it's unfortunate because you miss out on this holistic interaction that can happen when you truly put the soil food web at the keystone position, when you say that that's your primary focus. And so historic crop breeding has also really focused on making considerations for the environmental conditions. Think about all of the breeding programs that have focused on drought tolerance, so that when drought occurs in the environment, the plants are still able to be productive. Or the breeding programs have delivered management options to the food producer. So think about breeding programs that have created a consistent height of crops so that combines can more effectively harvest the commodity. And I would propose, putting this all together, that if we brought in hologenome breeding and really focused on the entirety of the soil food web, those important mutualistic partners, but everything else that's going on in that community in the root system of our crops, that the soil food web will help us take care of environmental challenges and will help us manage our agroecosystems. So the point of leverage, the place that we can have the biggest effect, is to bring in breeding that will really improve the outcomes of the soil food web. So I want to end with an optimistic thought, or at least it's optimistic from my point of view. I very much agree with this quote at the bottom of the slide, because agriculture is uniquely positioned to have an influence on our environment and on our society at the same time. And we have to get it right. In the next 30 to 60 years, if we do business as usual, we are going to see huge problems generated by our food production systems. We already see huge problems generated by them, but they're going to compound and really be a threat to our way of life. But if we take on this perspective, I think it will help. The picture above is one snapped by the Cassini probe as it was passing Saturn. It looked back past the rings of Saturn and it took a picture of that section of the sky. And that bright spot there in the sky is Earth. It's you, it's me, it's everyone that's ever lived. It's everybody that's a leader and has power in the world today. It's everybody living in poverty. It's children, it's adults, it's everything. It's our entire species. We live on one planet. And in the cosmic scale, we are small. This place where Dr. Elaine's Soil Food Web School is located, where all of these problems in agriculture are being grappled with, is really not too big to imagine changing. That together we can build a movement to regenerate our soils and to nourish this growing human population. And that gives me a lot of hope, because together we can do it.